afternoon, everyone. Hi. Hello. Hi. I'm Lynn. I am with the Coalition for Life Sciences, and I'm very grateful you all are here today. I'd like to welcome you to today's presentation of the Congressional Biomedical Research Caucus. To start, I'd like to thank our co-chairs of the caucus, Representative Steve Stivers from Ohio, Representative Joe Barton from Texas, Representative Jackie Spear from California, and Representative Steve Cohen from Tennessee for their commitment and dedication to the caucus. The Coalition for Life Sciences has been hosting these monthly caucuses for over 26 years now, but it is because of the commitment and dedication from the chairs of the caucus that we are able to host these briefings and opportunities for you. So today it is my distinct pleasure to introduce Dr. Denise Montell. Dr. Montell earned her BS degree in biochemistry and cell biology at the University of California, San Diego, and her PhD in neuroscience at Stanford University. As an American Cancer Society postdoctoral fellow at the Carnegie Institute, she developed a new model for studying cell motility in vivo using a powerful combination of cell biology and molecular genetic approaches. In 1990, she attained an independent junior faculty position at the Carnegie, and in 1992 became an assistant professor at the Johns Hopkins School of Medicine, rising to the rank of full professor in 2002. At the Johns Hopkins University School of Medicine, she served as director of the graduate program in biological chemistry for 10 years and was the founding director of the Center for Cell Dynamics, an interdisciplinary group of investigators from 8 different departments who shared a common interest in observing and manipulating biochemical events and cellular behaviors in real time in living cells, tissues, and organisms. She recently joined the faculty at University of California, Santa Barbara to pursue her interest in research, teaching, and program building. Today, Dr. Montell is here to discuss her breakthrough research. She and her team discovered a surprising reversibility of the cell suicide process. This understanding is vitally important. There's so much we need to know to understand about the life and death of cells in our bodies to truly move towards treatments and cures for diseases that afflict so many of us. So today, please join me in welcoming Dr. Montell.
Well, thank you. Thank you for the invitation, and thank you for coming. Every single day, every one of the trillions of cells in your body makes an active decision as to whether to live or die. And if cells survive that are not supposed to, we can end up with diseases like cancer or autoimmune disease. But of course, and so organisms have evolved mechanisms whereby cells commit suicide, and this process is known as apoptosis, in order to eliminate dangerous cells. But of course, excess cell death is dangerous too, and this can lead to degenerative diseases, the very widespread ones like Alzheimer's and others, and it can also lead to the dire consequences following heart attacks and strokes. So obviously, cell survival mechanisms are really important too. And so I want to tell you today about a novel cell survival mechanism that we've discovered and studied in my lab, and that we've named anastasis, which is the Greek word for rising to life. So originally, the process of programmed cell death, or apoptosis, was described as a series of very obvious, visible changes that cells undergo in a systematic way as they commit suicide. It's clear that the cell, if this is the outline of the cell body shrinks. The membrane makes all these folds, which are called blebs. The nucleus condenses and then breaks apart. All the organelles inside the cell break apart, and then the whole cell breaks up into these so-called apoptotic bodies before they're actually consumed and eaten up by other cells in the body. Now, that was our state of knowledge back in 1972, but we now know that those changes in the shape and behavior of the cell are driven by a series of biochemical events inside of the cell. And most importantly, there are protein enzymes called executioner caspases. And these executioners, as their name suggests, their job is to go around the cell and tear up all of the bits and pieces of the cell into little pieces. So these enzymes called caspases are very important because they break down the molecular components of the cell And that leads to these changes of the cell breaking apart. So in the field of apoptosis research, it's widely believed that once those enzymes, those execution or caspases are activated, and you start to see these changes in the shape of the cell, that this program is inevitable, that it's irreversible and rapid, and there's nothing you can do to stop it. However, a former postdoc in my lab, Hogan Tang, questioned that assumption, thought that it hadn't really been demonstrated very conclusively, and he did a simple experiment and got a striking result. So he wanted to test, is it really the case that cells that achieve this late stage of apoptotic cell death can't reverse that process and survive? So here's an experiment that he did. He took this is a primary liver cell from a mouse. That means a cell just taken directly out of mouse and cultured. We're looking at the outline of the cell here in this type of image. And then in blue, we can see the nucleus is stained, the DNA inside the nucleus is stained. And in red, one of the key organelles inside of the cell that's very important in life and death, the mitochondria, are shown in red. And now we're looking at a series of time-lapse images taken as this cell progresses. And at this time point, what Hogan did was to add 4.3% ethanol, and that's a lethal stimulus to the cell. And the cell undergoes the classic signs of apoptosis. The cell shrinks, the membrane makes these folds or blebs, the nucleus condenses, the mitochondria change, and if you leave the cells in this type of toxin, then they all die. However, if you do the relatively simple experiment of withdrawing this medium and adding fresh medium that does not contain ethanol or any other toxin, then something remarkable happens. After just 20 minutes, the cell looks a little bit better, and after an hour, it looks completely normal. And these cells will go on to survive and divide. It's not just that one cell or cell type. This is an entire field of human lung cancer cells stained in the same way. And we're watching them as they're treated to induce apoptosis, and then fresh medium is added, and the cells uniformly recover. So we've observed this in many different cell types, dozens of cell types, both cancer cells, such as you see here, and normal cells. And we've seen it in response to multiple different types of toxic inducers of apoptosis. What's remarkable is that part of the process of programmed cell death or apoptosis involves damaging the DNA of the cell, breaking it up into pieces as the cell dies. And what we found is that the cells can recover even after they have sustained this damage to their genetic material. This is an assay that detects DNA damage, and without going through the technical details, this is a nuclear stain, and it's staining for the DNA in the nucleus inside of a cell, and you detect these smears or tails, comet tails, when the DNA is damaged. So you can see that the treated cells, the apoptotic cells, showed signs of DNA damage. However, when the cells are allowed to recover, they repair that damage. So this is a cell survival mechanism. It's a mechanism that allows cells to recover and bounce back from the brink of death, and yet it also has the potential to cause mutations if not every single bit of damage to the DNA is repaired. So most of the DNA damage is clearly repaired, but we found that in a small fraction of cells that undergo this process, mutations remain. And here, what you're looking at are chromosomes from mouse cells that have been taken through this process. If the untreated cells, the chromosomes look perfectly normal, as they should, but if we treat 100 cells with an inducer of apoptosis and then allow them to recover, in 1 to 2% of the cells that recover, we can see obvious abnormalities in the chromosomes. Now, even in an even smaller fraction of cells, to 20 out of 10 to the 7th, or 2 out of 1 million cells, we will see signs of conversion to a cancer cell, so of oncogenic transformation. But we don't imagine that this process of cells being able to recover from the brink of death, we don't imagine that that process evolved in order for anyone to get cancer. That wouldn't make much sense. Maybe we imagine that it evolved for a beneficial purpose. So can this recovery from the brink of apoptotic cell death actually serve a beneficial purpose? And if you think about it, what would be really beneficial would be if cells that are difficult to replace in your body had the capacity to bounce back from the brink of apoptotic cell death. So take, for example, the neurons in your brain. We know that those cells are really hard to replace. Or the muscle cells in your heart. Those cells are post-mitotic cells. That means that they don't divide anymore, and so they're hard to replenish. So we looked at whether cardiac myocytes, that is the heart muscle cells, isolated from newborn rats, would have the ability to undergo this process of recovery from the brink of cell death. So in this experiment, we isolated cardiac myocytes from newborn rats, and we're following, in the different colors here, we're following 3 cell shape changes that occur during apoptosis and 1 molecular marker, 1 tag of an apoptotic phenotype. And what we can see here is that the untreated cells don't show these hallmarks of apoptotic cells. The treated cells do show the hallmarks of apoptotic cells, but when we withdraw the toxin and allow the cells to recover, they lose all of these markers of apoptotic cell death. So cardiac myocytes, or the heart muscle cells, also have the capacity to bounce back from the brink of death. So apoptosis, this word was coined back in the 1970s from Greek roots. It's derived from Greek roots that mean falling to death. And so we've employed the name anastasis to refer to this new process that I just showed you. And anastasis is an actual Greek word that means rising to life. So these observations, these striking observations, raise a number of interesting questions. We'd like to know where and when does this actually occur in animals. We'd like to know, including humans, we'd like to know what are the molecular mechanisms? How do the cells accomplish this great feat? And then can we acquire the ability through that knowledge to enhance this process or suppress it, depending on the situation? So let's start with the first question. Where and when does anastasis occur in animals? The trick here is, how are you ever going to know? Because the cells at the end of the process look very much like the cells at the beginning of the process, right? So if you simply open up an animal and look at the cells, you can't tell if they've had this life experience or not. So we wanted to develop a way of permanently labeling cells, marking cells that have undergone this transient near-death experience. So we developed a genetic method for doing this, and to do this we turned to the fruit fly, Drosophila melanogaster, which is a model that we've used a lot in my laboratory, because the genetics is very rapid and easier to manipulate. And so the conceptual idea that we used here was to convert this transient execution or caspase activity into a permanent genetic change in a cell that would show up to us as permanent expression of a fluorescent protein in that cell, if and only if it had gone through this transient near-death experience. So this is just a selection of tissues from a fly. We've made transgenic flies that allowed us to do this. And what we found was a very surprising result. We found evidence of widespread cell survival following activation of these execution or caspases in the animal. And without going through the details, so we saw that, in part, we saw that as part of the normal developmental process in the animal, that some cells go to the brink of cell death and then recover. In addition, remember, these are the mammalian cells, the human lung cancer cells that I showed you, where we induced them to undergo apoptotic cell death by applying a toxin. And we wanted to know, in the fruit fly, in the living animal, could we induce programmed cell death by applying a toxin and then see evidence of the reversal? So this is a picture of an ovary taken out of the fruit fly, and this was a tissue in which we did not see very much in terms of activation of these caspases without any particular stimulus. And we're going to look at a portion of the ovary that looks like this. And so what we found was that if we treated actually the whole flies with ethanol, so flies will actually drink ethanol, and they kind of behave like people. They drink, they kind of stumble around, and then they pass out, but they wake up again later. And what we found was that in the ovaries of the flies that were exposed to ethanol, but in very few examples in flies that were not exposed to ethanol, we saw evidence of this cell survival following execution or caspase activation, or anastasis. So what this technology in the fruit fly has allowed us to discover is that there's widespread cell survival following caspase activation, and we see three different kinds. We think we see caspase activity that's not really associated with death programs, but we also see anastasis, or recovery from the brink of cell death, during normal development, and then we can see this stress or toxin-induced cell survival. So another interesting question is, what are the molecular mechanisms? How do the cells manage to recover from the brink of cell death? And we're just starting these studies, and they're not published yet, but what we've done is to collect cells at various time points along this induction of cell death and recovery from death. And then we collect the cells and analyze the molecular components inside of the cells and say what's different and what's changing throughout this death and recovery process. And we have data that we're just now interpreting from those types of experiments. But what I want to share with you today really is our excitement about what we think the implications of this discovery are for human health. So we propose that anastasis may be a mechanism to preserve cells in your body that are difficult to replace. We believe that cells have this capacity to bounce back from the brink of apoptotic cell death in order to preserve them and limit the amount of permanent tissue damage that you would have in response to a transient injury or a transient toxin exposure. And so, for example, we're interested in testing whether neurons can undergo this process, and I showed you that cardiac myocytes can undergo this process. And work from another lab demonstrates that actually cardiac myocytes can recover from the brink of cell death after execution or caspase activity in response to transient ischemia events, so transient loss of blood flow to the heart. So, of course, this would be very important because that means that if we can come up with treatments that enhance anastasis, this could potentially be neuroprotective or cardioprotective, something you could give to someone before, during, or after a heart attack or a stroke that would limit the amount of permanent tissue damage that you would have from that. We wonder if the capacity of cells to undergo anastasis might change with age, and that's something that we're currently testing in the lab. Because if that were true, and we had treatments that could enhance this process, then this could be regenerative. But there's a flip side to this process, right? I told you that when cells undergo the process, there's a chance that they'll come out with a mutation, and there's a chance that that mutation will cause cancer. It's also possible that this is a mechanism by which tumor cells escape from chemotherapy. So we know that chemotherapy agents, chemo and radiation therapy, the way they work, the way they kill cancer cells is that they induce this form of cell death called apoptosis. And they are given transiently because they're toxic. And so we wonder if one of the reasons that relapse is so common after treatment with chemotherapy is because some of these cancer cells are bouncing back from this brink of apoptotic cell death. And if that's the case, then treatments that inhibit anastasis could improve the effectiveness of chemo and radiation therapies. So another line of research that we have in the lab is to identify small molecule chemicals that can either enhance or inhibit the ability of cells to undergo anastasis. So this discovery actually opens up a whole new field of research where we're interested in the basic biology and molecular and cell biology of this process. It has implications, as I mentioned, for regenerative medicine, ischemic diseases, degenerative diseases, and cancer, as well as developmental and stem cell biology and evolution. And I'll stop there and take your questions. Oh, actually, you know what? I'm not going to stop there. I'm going to, I want to acknowledge the people in my lab because, of course, this work gets done in a laboratory and it's a very collaborative effort. So Hogan Tang is the person who first discovered the ability of cells to bounce back from the brink of death. And this is the team working on this now, a PhD student, Austin Ding, a postdoctoral fellow, Gongping Sun, Yabdar, and Kelvin, who are both technicians in the lab. And I should also acknowledge the funding that supports this work. although this was when this project first got started in my lab, we had no funding for it whatsoever for 4 years. And then finally, we had enough preliminary data to talk a number of different agencies into funding it. And now we have generous support from the National Cancer Institute in the form of a Pioneer Award, from the National Eye Institute, the Keck Foundation, and a pharmaceutical company, Pharmacyclics. And that's the view from our lab. So now I'll stop and take your questions. Yes?
Molly Fishman with at here, one of the recruiters, and also a Santa Barbara grad, so both of us.
All right.
Anyway, we were talking about regenerative possibilities. Could you see that maybe even applying in like cases like Alzheimer's?
So I guess there are a couple of different ways it might apply in Alzheimer's. There certainly is apoptotic cell death of neurons in that disease. These are, the immediate causes of apoptotic cell death are different from what's shown here. But if we can learn enough about the molecular mechanism by which cells recover from the brink of death, then we might be able to manipulate it, and it could potentially be of use in Alzheimer's. Mm-hmm.
Quick question. You guys at Santa Barbara have a really great physics institute, the Cobley Institute, and also you're mentioning some stuff with neuroscience. And we know there's the President's Brain Initiative. So, twofold question: are you any role you're trying to play with the Brain Initiative? And then also, are you guys working a lot with modelers? Either mathematical modeling or quantitative modeling?
Uh-huh. So the second question first, in terms of mathematical modeling, there's a whole other project in my lab where we study how cells move, and in that case we work extensively with mathematical modelers because we know enough about that process that we can begin to take that kind of approach. I think we know too little here to start to model anything. We really are at the beginning of this brand new field, and we need to know a lot more before we could really move in that direction. As far as the Brain Initiative, yeah, sure, we have a Brain Initiative project ongoing. I just spoke actually not that long ago to a group that was planning the Santa Barbara effort towards the Brain Initiative. We've just hired some new neuroscientists on campus, and the first order of business for us with respect to whether or not this has anything to do with neurobiology is to ask, can neurons actually undergo this process of anastasis? We know glial cells do, so there are more glia in your brain than there are neurons, and those are support cells, and we know they have the capacity to undergo anastasis. We don't know yet if neurons do. We do see evidence in the Drosophila system of neurons undergoing this process, so we do believe neurons do have the capacity for it. And so, yes, I think that this work certainly could fit into the Brain Initiative.
Could you speak more on the mutagenic properties of the anastatic process, and how, coming back to the brink of cell death, what the percentages are, or
Sure.
what kind of cancer-related
Yeah. So the observation is that about 2%, 1 to 2%, depending on the cell type, of the cells that we take through this process end up with some kind of chromosomal abnormality. And perhaps more of them have some more subtle form of mutation. However, most of those are probably completely innocuous mutations that don't harm anything. And you think about it with a cell like a cardiac myocyte or a neuron, where it's it's not, those cells can't divide anymore. You may really be a lot better off saving that cell a little bit damaged than not having it at all. And so saving the cell and having it be a little bit damaged may not be so bad. In cells that are still dividing, this is a more dangerous thing for the body. But what we see is when we do assays for converting into a cancerous type of cell, then what we see is that it's really only about 2 out of 1 million cells that acquire that ability. So that means that in the vast majority of cases that these cells are recovering from the brink of death, they're just fine. But in a tiny, tiny fraction, they might acquire the ability to become a cancer cell. And the problem is that over the course of a lifetime, with the trillions of cells that you have in your body, eventually it only takes one cell to create a cancer. So it's a low probability event, and yet one that could have very dire consequences. But that's why we think that this process is mainly a beneficial one, that cells evolved the ability to bounce back from the brink of death, to repair tissues in response to injury, to limit the amount of permanent damage you would have in a tissue in response to some kind of transient event, toxin exposure or radiation exposure. And it's a sort of unfortunate side effect of this otherwise beneficial process that you could also end up with a mutation that would cause cancer, or the ability of a cancer cell to bounce back from chemotherapy. Did that answer your question?
Hi, this is really exciting, and it also sounds like it's really early. And probably what everybody wants to know is, are therapies like 10 years, 20 years, are they like way down the road in terms of cancer and restoring parts and neurons?
Right. So, the nature of research is you never know where the next breakthrough is going to come from, and it's really hard for me to say what's the timeline. I mean, we get up and go into the lab because we think every day we might find it. If we do a small molecule screen for things that can enhance or inhibit this process, that could take a year, and if we got a really great hit, then we could move forward with it more rapidly, and maybe the benefits would come sooner. But in research, you never know what obstacles you're going to encounter, what challenges are going to be raised. You think you can get that screen done in a year, and then this, that, and the other thing happens that you weren't anticipating, and it takes 2 years instead of one. So I'm sure I would love more than anybody probably to be able to tell you exactly how long it would take for this to translate into an actual therapy for somebody. And the fact is, we just don't know, and that's the nature of research.
Have you tested this idea? You showed us normal cells very convincingly, but have you taken, for example, cells that have the mutations characteristic of Parkinson's disease, where you lose a specific class of dopamine-producing neurons? Have you tried to see if you can take a diseased population of cells, treat them in this manner, and rescue their eventual viability?
Yeah. So no, we haven't, and like I said, we still don't know if, let's say, the dopaminergic neurons themselves, even before they're diseased, we don't know if they're capable of undergoing this. So that's the first thing, and there's someone working on that right now, taking different parts of the brain and asking which types of neurons can or cannot do this process. Once we know that normal neurons can, then it would be interesting to see if, let's say, neurons in Parkinson's disease were compromised in their ability to do that. But again, it comes back to, we need our hands on molecules that will allow us to manipulate it, make it work better in the case of diseased neurons or cardiac muscle, and make it work worse in the case of cancer cells. That's where the therapeutic potential lies, I think. Yes.
You've already talked some about reviving brain cells that are difficult to replace, and I'm wondering if there's any implications for like brain injuries or brain damage, such as like an anoxic brain injury.
Yeah, exactly. That would be the type of situation where we would think there might be a benefit if we can do this. So as I mentioned, the best evidence out there right now in the literature is from one lab that showed that if you tie off the artery to the heart, not the brain, but to the heart, in a rabbit or a rat, you can see evidence of apoptotic cell death, and you can see evidence of those apoptotic cells recovering the next day if that tying off of the artery, that stopping the blood flow, is transient. So they did it for 5 minutes. If we, and that looks pretty solid. So if we extrapolate to the brain, if someone were to experience a loss of blood flow to the brain for a transient period of time, the cells might bounce back, and maybe that's what they normally do, and maybe that's why the damage from strokes isn't worse than it is. But we know that there are waves of cell death that follow that kind of an event. There's the initial loss of cells from the ischemia, but then there are waves of subsequent cell death that happen from the toxic soup that happens when the cells die. So it would be those second waves that we would hope to intervene and prevent, and thereby limit the damage. Yes.
So when you did your tagging experiment, I mean, how much is this revolutionizing like what we think of as normal programmed cell death? Did people know that this was happening in theoretically normal tissue, that throughout development this happened, and there were signs that some of these?
Yeah, so no, people didn't know this. And this is really a new way of thinking, and it's, I presented the work actually to a room full of experts on apoptosis, and yeah, it's surprising. The main thing that's surprising is how many cells end up showing signs of having been through this process. Because, so during normal development, some cells die by apoptosis. That's, we've known that for a long time. The reason you have fingers instead of a little paddle-shaped thing is because these cells die. And there are many other places. The brain is another place where some cells have to die. For whatever reason, the embryo produces lots too many of them, and then some of them die by apoptosis. It was not appreciated at all that cells could bounce back from the brink of death, but it might make sense. I mean, so in the brain, for example, the reason some cells die is there's a limiting amount of growth factor there. So there's some nutrients there and growth factors specifically that the cells are going for, and when they don't get enough of them, then they die. But you can imagine that being a transient phenomenon. There's millions and millions of cells, and the growth factor accessibility may come and go, and so maybe they're transiently deprived of growth factor and they start to die, but then they get the growth factor back and they bounce back. So it seems to be, we believe that our evidence is most consistent with the idea that sporadically in time and space, while apoptosis is happening, every now and then one of these events happens where the cells recover, and then all of their progeny go on to live and thrive, and that's how we end up with so many cells looking like they have been through this experience. But yeah, I think it does change the way you think about the process. But a very interesting idea that I don't know the answer to yet is whether they actually, if what doesn't kill you makes you stronger. Are the cells that come through this process actually somehow, better able to populate the tissue, and is that one reason why we get so many of them? Other questions? Yep.
Can you talk some more about do you have any theories about the molecular underpinnings that you kind of?
Mm-hmm. So I glossed over that, not wanting to give you lists of genes and put everybody to sleep. But so yes, so conceptually, I can tell you, based on new results, they're not published yet. I can tell you that what we see is that as the cells start to die, there are big changes, in their metabolic state, and that you would expect. As they recover, there's a very instantaneous or nearly instantaneous burst of gene activity. And this gene activity looks very much like an immediate response to growth factor. So they're coming out of their, when they're dying, they're shutting down all the growth activities, and when they're coming back, they're reactivating all those growth activities. But when they're dying, they also activate a stress response. And when they recover, they're still activating the stress response. So as opposed to normal growth, the recovery involves both a stress response, a growth response, and then in addition, the cells secrete factors that attract blood vessels to them. So you can imagine if you're a cell in a tissue and you've just gone through this near-death experience, you probably imagine some of your neighbors actually died. You bring in the nutrients, you call in the nutrients, so they secrete factors that draw blood vessels to them and dilate the blood vessels. But what's really cool about this is that it's a two-step process. So that's step one. The next thing they do is they divide. And so again, you might imagine that if you're in a tissue that's just been damaged and some of the cells have died and some of the cells have lived, the ones that have lived want to divide to make up for the ones that died. These incredibly beneficial responses in a wound healing situation are the worst case scenario in a cancer situation, right? So the cancer cell may be doing something that it evolved as a wound healing response, but now doing all those same things, calling in the blood vessels, bringing more nutrients, activating growth, and activating proliferation and division are exactly the worst case scenario for the cancer cells. But that's what our preliminary evidence on the molecular pathway looks like. Yes.
This might be a little bit speculative, but do you see any potential applications for either life extension or performance enhancement from this line of research?
Oh, wow. Okay. Life extension. Well, I hadn't really thought that's a little out there for me, but I hadn't really thought about that. The whole field of aging research, I mean, I think most people are really more interested in enhancing the years that we have than necessarily significantly extending them. There are a lot of issues with what would happen as a society if that happened. But I do think that this has it's possible that cells lose the capacity to bounce back. As we age, maybe our cells age and they lose the capacity to bounce back from the brink of death. And if we could enhance their capacity to do that, then maybe they would continue to keep us younger for longer. But I'm not sure in terms of life extension, other than, let's say, preventing death by preventing cardiac, death from strokes and heart attacks and cancers. I guess that's a form of life extension. But if you mean normal life extension, I haven't really gone there thinking about that. And performance enhancement, no.
Are there implications to this as well? I'm thinking particularly in terms of if you can suppress the regeneration for cytotoxic T cells or something like that that are using these mechanisms to kill infected cells. Has that been explored at all? Is it a reasonable extrapolation or no?
It's certainly an interesting thing to think about, and we haven't worked very much with immune cells. We have seen the one macrophage cell line that has the capacity to bounce back from apoptotic cell death. Their cell death plays an enormously important role in the immune system, so it would be shocking if it didn't have some implications for the immune system. The interesting point that raises, I think, is that one of the things that we're working very hard on right now is trying to figure out, is there one shared pathway that all cells use to do this, or do different cells have slightly different versions of the mechanism? And if they did, that would be really useful, because you would then have the capacity maybe to target it more specifically to brain cells or immune cells or the cytotoxic immune cells versus the regulatory immune cells. So I think that that's something that we're very interested in, is figuring out which aspects of this program are just conserved between all cells and which ones are different for different cell types, so that we can potentially gain control of it a little bit more. Did you have a question?
I think it's, when you're trying to kill cancer cells, it's kind of tricky to, because the healthy cells are also killed. And then it's kind of tricky which one should go back, because you don't know.
Right.
But when somebody had an accident, for instance, and they get oxygen, and the brain cells are working, that doesn't have anything to do with cancer cells. So that can be repaired. And for new cells, so
Yeah. No, so that's a good point, and it comes back to what I was just saying about trying to understand whether different cell types achieve this survival with different mechanisms or the same mechanism. And of course, we hope that cancer cells do it a little bit differently than normal cells do, so that we can find treatments that will inhibit the ability of cancer cells to do it, but treatments that will enhance the ability of normal cells to do it. And there are many opportunities for that to be the case. Maybe cancer cells and normal cells have very similar mechanisms. Maybe they have different mechanisms, but also maybe, lung has a different mechanism from brain, and if you have a lung cancer, you can, target the lung mechanism, and if you have a stroke, you can target the brain mechanism. So this is all, I mean, speaking of speculation, this is all speculation right now, because we've actually only got our first glimpses into the molecular mechanism for, in fact, cancer cells. So we need to go back and do those same experiments on normal cells, and especially, yes, I think there's a lot of interest in neurons.